It is my privilege to start the next session by introducing our moderator, Professor Rick Huganier from Hopkins. As I mentioned at the top, he's one of the first people we spoke to. He's one of the first grants we gave. It was his lab in 1998 that identified the SYNGAP gene. And he's got an incredible bio, and you should read it online. But please give your attention to Rick for this super important session. And I'm not going to do it, but I'm going to ask Rick if you want to just remind people how important animal models are as we go from these ideas to drugs in kids, just I think so the families can get it. Thank you. Professor Huganier.
Well, thanks, Mike. It's really a pleasure to be here and honor. It's really been incredible honor to be involved in SRF, and also I want to thank for the funding as well, but also to be involved with families and meet all of you. We've made several mouse models based on some of your children's mutations, and so we actually introduced some of the kids to their mouse model, and so we also have a clinic working with Connie at Hopkins and KKI to see the kids, and so we've had a lot of interactions with the families as well. So you're all heroes in my mind. It's really impacted the motivation of people in my lab too. Ingi Hong is here today. Just seeing how the families and knowing what you're going through, that it's really motivated us tremendously. Being basic scientists, we were interested in basic mechanisms, but this definitely gives an extra boost in our motivation. So animal models are really key. I'm a great believer in animal models, including mice and zebrafish and other models. I think with SYNGAP it's actually particularly important because the mouse mutations actually model children relatively well. They have learning disabilities, they have sleep problems, they have seizures, and so I think that they're a fantastic model. And we've made the first one of the first knockouts of SYNGAP heterozygote, back in 2000. And that mouse has all these behavioral and deficits that we now associate with the kids. So we think it's a great model and to test a variety of therapeutics on. So today we're going to focus on some preclinical models, and it's a real honor to introduce the first speaker, Gavin Rumbaugh from University of Florida Scripps, and particularly because he was a postdoc in my laboratory. So I'm very proud of him. He's come a long way since being a postdoc in my lab, and has been a central player in the SYNGAP story. So, Gavin.
Good afternoon, everybody. Hopefully everybody's well fed now and refreshed from the amazing morning session. Can you hear me okay now? It's okay? Great. And so I echo everything that Rick said. In fact, I'm a cell and molecular neuroscientist by training, but I'm going much more towards understanding of, as you'll see, neural circuits and behavioral regulation, in large part for two reasons. One is because the SYNGAP model is an outstanding model. They're not always this good and reflective of some of the domains that you see in human genetic equivalents, but the SYNGAP model is great, and it's inspired me, along with meeting many of your children, to understand. What I really want to do in my lab is to understand the neural basis of behavioral dysregulation associated with single gene Mendelian neurodevelopmental disorders. And almost all my ideas have been generated by unexpected brain function changes in the SYNGAP model. And so I plan to keep studying this model because I keep discovering what we think are new and interesting things in the model. So first, I'd like to just give credit to people in the lab. So I I write and talk on the phone and come to meetings, but these people do the experiments, and without them, we wouldn't be able to talk and share data. So, and today most of what I'm going to show you was by this postdoc Vineet Arora, and also a former graduate student, Murat Klinik, who I guess this is a very recent picture, so Murat's not on here. So thank you very much. So I'd like to tell you what I'm going to tell you before I show you, just to maybe hopefully pique your interest. And so today, I'm going to tell you about a novel function of SYNGAP1. Don't worry, Mike. I'm going to talk about isoforms, and so the isoforms are helping us understand novel functions of SYNGAP1 at the whole animal and the whole brain level. And so, what this novel function is we view it as the gene is of course a major synapse regulator, and that's still true. But Spectrum News had an article recently about genes doing double duty. And so I think SYNGAP is going to be an archetypal representation of this is that one of the reasons why I believe that when you lose one copy of it, it becomes a disease gene. This is very rarely the case. The reason why is because SYNGAP does double, triple, perhaps even quadruple duty. So it has many, many roles, and not all of them are in the synapse. And I'm going to show you one that we've recently discovered. And this function is uniquely developmental, and it controls how the brain organizes itself, how the brain creates what are called neural circuits and neural networks to be able to control behavior and thought. So I believe it's very relevant given some of the clinical talks we heard this morning, which is where the behavioral dysregulation in SYNGAP is clearly one of the challenges that face Syngapians and their parents. And I mentioned this already, the studying these isoforms, and I'll get into what we mean when we say isoforms, is actually helping us to tease apart these what we call pleiotropic or multiple distinct functions of the SYNGAP1 gene to mediate changes in the brain. Okay, I'm not going to have too much media today, so this is maybe one of the only videos, and so our brains are wired together, and within this neural network is how information is transferred from neuron to neuron. And when enough neurons in the right place fire at the same time, that's how thoughts emerge and decisions are made. And so the idea is that what SYNGAP1 DEE is, at the most basic level, is it's an alteration of brain circuitry that occurs during development. So these pathways for neural activity are not constructed in the way that you would see in a neurotypical child. And now what happens when you have abnormal wiring in the brain is that electricity has a hard time in some cases transmitting itself around the brain, which means that that will result in altered behavior and altered intellectual ability. And so ultimately what this does is it impairs what we call internal brain processes. These mysterious processes in the brain that emerge when neurons fire together that allow for behavior and thought. And so one of the goals of modern neuroscience is to use animal models and to understand what these mysterious internal processes are. And so what we believe is that studying these disease-causing genes like SYNGAP1 will speed up the process of understanding this mysterious process because we know that they're altered in the human population. Okay, so I'll try to stop this so I don't make everybody dizzy. Okay, so I'm going to try, so one thing I wanted to say in my introduction is I'm going to try to thread the needle. So I want to be able for Rick to be interested and Liz and Sally to be interested, but I want all of everyone else to be interested and follow along too. And so I'm going to try to thread the needle. I'm going to show some data, and I'm going to raise what we think are new concepts, but I'm going to explain them very generally. So please, my colleagues, don't give me a hard time if I gloss over things. The data are all there, and I'm happy to talk about any of the data after the session. So it's important to understand the brain works across different scales. So of course, neurons are connected to each other through synapses. And that's a chemical synapse, and that's how electricity is moved from one neuron to the next through the chemical synapse. And of course, we know that SYNGAP is a major regulator of how individual synapses can strengthen or weaken to store information, which is important for memory. But there's another process is that if you have, it doesn't matter how much synaptic drive or excitability that you have on a neuron, that can't transmit to the next neuron down the line if you have a dysregulation of what's called neural excitability. So these are various ion channels that sit on the membrane, and they basically regulate how easy or hard it is to make that neuron send the communication to the next neuron. And so, these are all the different places where genes can affect the function of information transfer, which is at the heart of a DEE. And so again, then neurons are connected and can fire at the same time, and then you get what's called ensemble dynamics across the whole brain, and then that leads to behavior and thought. Okay? So the idea is that we know that SYNGAP works in the synapse, and the work on SYNGAP thus far has been very synapse-centric. Of course, because of the way it was discovered, right? And it's in the name, so people just assume let's study in the synapse. I'm just kidding, but it's important to note that it's quite possible that it's now becoming not the exception, but the rule that these disease-causing genes that cause terrible neurodevelopmental disorders are actually working in multiple places across different times in development to change brain function. And so we've been trying very hard to discover new functions of SYNGAP1 because that will give us more information about how to create new therapeutics to be able to treat some of the most debilitating aspects of the disorder. And so to Rick's point, one of the reasons why this mouse model, which is a SYNGAP heterozygous mouse or a mouse with haploinsufficiency, one functional copy of SYNGAP, is that it has seizures. And it has seizures that look a lot like you see in kids. So here, I don't know if you can see the shading. So this is from a collaborator, Jimmy Holder, from a SYNGAP1 patient, where he sees spike and wave discharges, and he saw a high propensity of these at sleep-wake transitions. And so when we looked in the mice, and actually this is the mouse that Rick had created in 2000, and we look in this mouse and we see here, so these are EEGs and along with depth recordings, depth electrodes in the hippocampus. And in the shaded region here, this is a sleep-wake transition, and you see these, and this is a 1-minute scale. So you see lots of these spike and wave discharges in the SYNGAP mouse that only has half of SYNGAP1 expression. But this mouse we can do a fancy trick where we can induce and reactivate in the same animal, we can induce normal levels of SYNGAP. So we can do gene therapy in a mouse and we can restore SYNGAP expression. And what we see is that these what are called interictal spikes, spike and wave discharges completely went away. And so here's the example. This is the same mouse before and after. And so here you can see this would be all the spike and wave discharges before the treatment. And then you can see here that in every animal, all of those spike and wave discharges went away. In this same paper in eLife in 2019, we showed that this was also associated with improved long-term memory and also a change in the seizure threshold. And most importantly, this was in adult mice. So that means that restoration long after development was over improved predictors of seizure and also improved memory. And so that combined with the fact that we see that discharges at the same time tells us that whatever's happening underneath in the brain, we need to understand what's going wrong because we think that we have the right phenotypes to study. Okay, so if we want to understand SYNGAP1 function, so the gene encodes protein, as you heard this morning. But the gene can actually encode multiple different proteins. So through processes that I don't know if Liz is going to start ATGs or not, but you can have multiple different protein entities made from the single SYNGAP1 gene. And today, mostly I'm going to talk about alternative splicing in the end of the SYNGAP1 gene that can encode 4 different kinds of proteins that are 98% the same, but the very end of what's called the C-terminus of the protein contain somewhere between 4 and 35 different amino acids. So very small amount relative to the total. But some pioneering work from Rick's lab and also Mary Kennedy's lab had shown that we knew for almost 20 years now that these isoforms with these different C-termini, with different protein sequences, have distinct functions. But what we don't know is how these distinct molecular functions translate into changes at the whole brain level in animal functions. So what we had done was create a series of animal models that tinkered with the relative expression of these different isoforms to be able to see if that would help us understand new functions of the gene. And so one thing to think of is that when you have, and this would likely be the case in Syngapians, is that when you lose one copy of SYNGAP, you're losing one functional copy of the gene, but you're losing half of all of these protein variants. So if these protein variants have different functions, then perhaps you're losing, instead of one protein that's lost, you're losing half of one protein, you're losing half of 3 or 4 different proteins. And so that gave us the idea that maybe these isoforms are mediating different types of processes in the brain for different types of behavioral phenotypes. Another thing that made us think that these isoforms would be important for understanding new functions of SYNGAP is that they're very clearly regulated differently over space and time in the developing mouse brain. And likely the same would be true in the human brain. And so because they can be expressed at different levels, in different types of neurons in different parts of brains, if they had different molecular functions, which we know from Rick and others' work, that it would be very likely they would be affecting different types of neural circuits and therefore different types of behaviors or different types of memory. Okay, so the hypothesis is that the range of phenotypes in animal models arrive from distinct cellular functions of the SYNGAP1 gene. Okay. So we have molecular functions, which is important because that's what we drug, right? But then we have to parse those molecular functions into cellular functions. So synapse regulation would be a cellular function. That's something that happens inside of an individual neuron. And we know that one of the roles of SYNGAP in the synapse it's called a RAS-GAP protein. And so what it does is it activates signaling pathways that suppress the strength of synapses. And so when you lose one copy of SYNGAP, it's been shown that what you end up with is too much excitability in your synapse. The synapses are too strong. And that mechanism comes from, it appears to be an interplay between movement of SYNGAP in and out of the synapse, along with the fact that the physical protein of SYNGAP acts like a scaffold to help organize signaling in the synapse. And so when you don't have the protein, you don't have enough of it, so you don't have enough of it moving back and forth, but you also don't have enough of it to act as a scaffold, and that appears to dysregulate synapses, okay? But one thing that my lab had discovered, and now has been found by other labs as well, is that this also has a tremendous effect on what's called dendritic morphogenesis. Now synapses here, that's one synapse, it sits on a dendrite. And so if you have alterations in dendritic morphogenesis, so you have too much dendritic space or too little, you lost your scaffold for forming synapses. And so by definition, if you change dendritic morphogenesis, you're changing circuitry because you're changing the way, your ability to make new connections between neurons. And so SYNGAP does this in a very complicated way, and this dendritic morphogenesis phenotype, which regulates wiring of the brain, appears to have nothing to do with its role in the synapses, which is what I'm talking about today. And importantly, a lot of the dendritic phenotypes we found in the animal model are recapitulated in both human neurons derived from full knockout SYNGAP1 neurons. And we've also seen these dendritic phenotypes in several different patient neurons derived from patient iPSCs. So as like the seizure, we believe that the dendritic morphogenesis is also conserved across mammalian species. Okay, so what do I mean by complex dendritic phenotypes? So these are neurons that are traced. Okay, and so this is one brain area called the somatosensory cortex. This is where your brain codes sensation from your body. Okay, and it represents what's going on to your body in your cortex. So you have superficial neurons, which are a little bit smaller, and then you have these deep, large output neurons. And so during development, these superficial neurons are stuck in a state of arrested development. So they never grow to be the right size. So there's not enough excitation generated by these neurons because they never formed enough synapses. But just half a millimeter below that, in the very same area of the brain, if you see here, in these SYNGAP1 heterozygous mice, these deep, very large layer 5 neurons are fully mature almost a month before you would see in a wild type. So in the very same part of the brain, you have neurons that are growing too slowly, and just below that, you have neurons that are growing far too quickly. And so that's inverting the way that the circuits are built in this part of the brain, okay? Interestingly, both types of neurons have the same synapse phenotype, which is to be too strong. So that was the first clue that there must be some other biology going on to regulate dendrites that are distinct from the synapse. Okay, and I think I already went through this, is that we care about dendritic morphogenesis because if you want to wire up circuits between multiple neurons, you can only do this by growing dendrites because that's where synapses are made. Okay, so again, I'm going to tell you what I'm going to tell you first, and then I'm going to show you the data. So what I'm going to show you is that the way neurons normally grow their dendrites during development in the cortex, these are excitatory neurons, the neurons with spines that have a lot of SYNGAP, is that they're in what's called their intrinsic excitability, or their just general ability to fire action potentials, is governed by a delicate balance of voltage-gated and passive ion channels that set excitability on the membrane. And it's been shown by Mu-Ming Poo's group and others, and you don't know who Mu-Ming Poo is, but Rick does, that the way that these neurons fire in development is crucial. Those action potentials act as a trophic or growth signal to make them grow, right? So the neurons sense how much activity they have, and that makes them grow, okay? What we found in SYNGAP is that these particular potassium channels that regulate excitability are completely dysregulated, and what that does is in the sensory cortex is that's the mechanism for why those neurons are too small. And I'm going to show you some data to hopefully convince you of that. But interestingly, the exact same mechanism occurs in a different part of the brain, in the prefrontal cortex, which is involved in decision making. We see the exact same effect in that potassium channels are dysregulated, but they're inverted in the completely opposite direction. So this is the same mouse, this is the same gene that's disrupted, it's SYNGAP, two different areas of the brain, the exact same type of neuron, they're just separated by several millimeters in different parts of the brain. Here, instead of potassium channels being expressed at too high, the potassium channels are expressed too low. These neurons become hyperexcitable and actually end up growing too much dendritic field, okay? And so we're fascinated, how can the same gene, and I'll show you that this is autonomous to the SYNGAP function within these cells, how can the same gene regulate potassium channels in opposite directions to control growth in different ways? So when you're a biologist like me, that's like something that, you know, and then if you can associate that to something that might be going on in the brain of a child with a rare disorder, you know, this is incredibly exciting for us to study. Okay, so here's just some data. This is the part where I may go a little fast. I'll point out the important points. So this would be this idea of what we found in the beginning was this correlation between the way that neurons grow, their expression with SYNGAP, and how easy it is to fire action potentials, okay? And we also found that that was inversely correlated with the amount of potassium channels. And that's important because potassium channels make, the more potassium channels you have, the harder it is to make them fire action potentials. So if you have too many potassium channels on the surface, it doesn't matter how many synapses fire to make that neuron express an action potential, they'll be completely shunted. Okay, so these neurons in layer 2, 3 are very difficult to fire action potentials, as you can see here. So a certain amount of input, you get this many spikes. In SYNGAP1, you get far fewer. And here you can see that in everything that I'll be showing you, red is the SYNGAP1 heterozygous mouse, and blue is the wild type control. So we're always comparing the SYNGAP loss of function to his brothers and sisters that are in the same litter. Okay? So too many potassium channels, small cells, hard to fire. If you just go 3 or 4 millimeters to the prefrontal cortex, you see exactly the opposite, as I mentioned. So you see neurons that grow much faster, they fire way more action potentials, and they have far less potassium channel function right here. Okay. So, and this is important for the biologists in the group, is that it would be very easy when you see these inverted phenotypes to be like, well, it's some compensation in the system. There's something about the front of the brain that's trying to compensate for the back of the brain. So we do what's called cell autonomous to understand if this is an effect of is SYNGAP having opposite effects in two different kinds of neurons. So we're able to do that using another type of mouse line where you can conditionally disrupt its expression in just a few cells across the brain. So 99% of the cells are wild type, are normal, and then the ones that turn green have SYNGAP haploinsufficiency. And so that's what you would see here. Okay, and we convert these green cells. Hopefully you can see. Yeah, these few green cells there are haploinsufficient neurons before birth, and they stay haploinsufficient for their whole life. But everybody else, all of their neighbors, are wild type. And to make a long story short, we see exactly the same thing. This is the sensory cortex, but we see the exact same cell autonomous effect in the associational cortex, in the prefrontal cortex, where you see increased spiking activity. So we know that this is the function of SYNGAP in these neurons. So somehow it's having opposite effects. Okay. Okay, so next we wanted to understand is because there's this principle of homeostasis in the brain where excitability can regulate synapse function, and synapse function can regulate excitability. And so what we wanted to do is to really drill down, and is this really a novel function of SYNGAP working to regulate potassium channels out in the dendrites? Is it really something different that's going on in the synapse? So one way to do this was to use a molecular tool. It's called a dominant negative potassium channel subunit. And what that does is when you express it genetically, it acts as a sponge and it grabs potassium channels and pulls them into the cell. So you can, it's like a genetically encoded drug that decreases potassium channels, okay? So because we knew we had too many potassium channels and we thought that that was driving the small neuron phenotype, we said, well, what if we circumvented all this process by just using this potassium, dominant negative potassium channel to lower SYNGAP excitability back down. Okay, so we tested this tool by doing some techniques, again, where we can put this in just a few cells, and then we measure this. And to make a long story short, this dominant negative potassium channel was able to correct the potassium channel. It was able to lower potassium channels back to wild-type levels, and it completely restored excitability in these SYNGAP heterozygous neurons. So you can see where the arrows are. So this would be the SYNGAP plus the dominant negative potassium channel, and this would be the wild type without the potassium channel. So now that we have this tool where we can intervene and lower the membrane excitability, we asked ourselves, well, does this actually regulate dendritic morphogenesis and, more importantly, wiring of the brain? So we did this experiment. So we did another type of biological technique. This looks like it maybe took a couple weeks. This took 4 years. So it's kind of funny to give these talks in 10 minutes. Vineet, has spent. He spent probably 6 months just figuring out how to do this experiment. So, I'm very thankful for his hard work. And so what we can do is we can express these potassium channels, but then we can go in and harvest the brains from these animals, cut live acute slices, and then what we can go in is we can target a cell that has the potassium channel and a cell that doesn't, and then fill them with a dye, and then we're able to trace the dendritic arbors in these neurons. And so interestingly, in wild-type neurons that express the potassium channel, and we show in the previous slide that these wild-type neurons also become more excitable. But interestingly, this has no effect on dendritic morphogenesis. So that tells us that there must be, if the neurons get enough activity, they're good. They grow to the right level. Any more activity is probably okay. They're good. But what we found, the opposite in SYNGAP. So it worked the way that we predicted, is that when we make SYNGAP neurons more excitable, what we found is that we're able to improve the morphogenesis, and actually the morphogenesis is now, those neurons are the same size as the wild-type neurons. And so that really is a strong indicator that the overexpression of the potassium channels is driving too few spikes during development, and so these neurons are stuck in this arrested development phase. Well, what about wiring the brain? So not to belabor it, there's these neuronal connections that I talked about. There's many of them, but there's a way that this information in the sensory cortex is passed on from one place to the next. It's called feed-forward excitation. And so what we did is we stimulated neurons in what's called layer 4, and then we did a whole cell patch clamp recording where we can look at synaptic currents in layer 2-3, and when we stimulate in layer 4 and layer 2-3, we see these waveforms reflect how much synaptic connectivity is between those two types of neurons. And so when we express this potassium channel, we see in wild types, we saw no change in dendritic morphogenesis, we see no change in synaptic connectivity. However, we found an increase in synaptic connectivity in SYNGAP mice. So this is, I think, really good evidence that what SYNGAP, when you lose one copy of SYNGAP in these neurons, too many potassium channels go to the surface, they don't spike over development, and they have arrested development. But if you're able to go in there and intervene and lower those potassium channels, you can bring that connectivity back. Now, I haven't done all the Vineet hasn't done all the experiments in the prefrontal cortex, but it looks like the same story is true, except you have to intervene in the opposite way. So you have to express more potassium channels, not less. And so that means that one might think, well, let's just give a potassium channel inhibitor to these mice and you might make everything better. But what we're learning, and this is why you have to study animal models, is you see complex phenotypes in different parts of the brain. And so a potassium channel inhibitor might be great for sensory cortex function, but it would be terrible for the prefrontal cortex, or the area of the brain that helps you make decisions. And so you would need a targeted therapy for both types of these neurons. The best targeted therapy, in my opinion, would be to put the protein back in, because we don't know what the protein is doing in the sensory cortex versus the associational cortex. We're doing it, and we'll figure it out eventually, but the protein does it. That's one thing we do know. And so that's why I'm a big proponent, and we'll support anyone, and we'll collaborate with anyone who has an interesting way to try to raise SYNGAP protein, because I believe that the more I learn about the complexities of the model, it's really important. Neurons really want their SYNGAP back. I think that's what we're learning. Okay. I mentioned this chicken or the egg problem, and so it's kind of hard to define especially with such dogma in the SYNGAP field, and it's dogma appropriately. It is a major synapse regulator, but the problem is when you go to submit papers or you go to submit reviews, whatever you're seeing at the synapse, or whatever you're seeing in dendrites, that's because the synapse is doing everything. you have to spend a lot of time to really prove that it's a different function. And so what we did is we did this experiment we did what's called homeostasis. So as I mentioned, that if you decrease excitability of a neuron, neurons want to fire a certain number of action potentials over a certain amount of time. Okay? It's called a set point. It was discovered by Gina Turgiano. And so if you make neurons artificially fire too many action potentials, they'll reduce their intrinsic membrane excitability to stop that from happening. And if you make them fire too few experimentally, they'll do the reverse and they'll start firing more. But synapses will also do the same thing. Synapses will strengthen or weaken, right, independently. And those have divergent mechanisms, okay? All right, so what did we find in these SYNGAP heterozygous models? Well, interestingly, and the reason why we did this experiment is if it's true that intrinsic excitability is really the driver here, we would, and these neurons are stuck in this low excitability state, if we made them fire more action potentials, the hypothesis would be, well, they wouldn't be able to bring up their excitability because they're stuck, if it's pathological. So in wild-type neurons, you can see that when you give tetrodotoxin, which inhibits spiking in neurons and has been shown for 25 years now to increase neuronal spiking through changing membrane excitability, you get this really nice increase. That's our control. But as you can see here in the SYNGAP mice, there's no self-tuning of membrane excitability. It's completely gone. So that explains that when these neurons are born in development, they are stuck in this excitability state because they're not able to tune themselves back up. But interestingly, we did synapse scaling, and this was a synapse, as we expected synapse scaling to be blocked here as well. However, what we found, at least in the heterozygous mice, is that the synapse's ability to scale themselves up was intact. So at least in the heterozygous mouse, we have a larger effect on the intrinsic membrane excitability self-tuning than the synapse. So this is more evidence that it's living outside the synapse. I took this nice model from one of Rick's papers from Rocky 2020, and they have a model based on different isoforms. So this alpha-1 isoform, which I think I'm a big fan of, I think it's a really important isoform. It's this protein variant that seems to live pretty much in the dendritic spine synapse. I think most of us agree that's where it does its job, okay? Some could argue that nature gave us that isoform so SYNGAP could do its jobs in the synapse, okay? But there are three other important non-alpha-1 isoforms, and Rick has shown, and we've shown as well, that those live much more in the dendrites. They're still in the synapse, but they're also in the dendrite. So the idea is that maybe these non-synapse isoforms could be the smoking gun for regulating potassium channels. I don't have that answer right now because we're doing those, and that's a little bit more of a complicated experiment. But in this paper that we published recently, we did make a mouse where we were able to disrupt functionally the alpha-1 subunit. And we show that those animals have cognitive impairment and they're susceptible to seizure, and I'm going to show you some of that data a little bit later. But we thought that this would be a really good test for the hypothesis that these neurons would have normal intrinsic membrane excitability tuning because they didn't lose their dendritic isoforms. They lost only their synapse isoforms. And this is published. You can show that this mouse does what we expect. We don't change the expression of the isoform, just its ability to do its job by binding to its partners. And so the experiment worked in the sense that the self-tuning in this what we call PBM mouse is perfectly normal. They're able to now tune. So if you lose all of the function of this alpha-1 isoform in terms of its binding capacity, which has effects in the brain on behavior, it doesn't appear to do anything to its ability to self-tune its membrane excitability. However, as expected, the synapse tuning is gone. And that's not surprising given that we know this is the synaptic isoform. Okay. Now, since we have this really nice, the expression of the potassium channel dominant negative showed us that the mechanism for the small neurons appears to be the regulation of the potassium channel and the tuning of membranes. So given in these alpha-1 mice, these PBM mice that have normal tuning, the hypothesis would be, well, they are probably firing normally during development and they shouldn't have altered dendritic arbors. And in fact, that's exactly what we found. The arbors, if anything, are actually slightly larger. And we think that's because their synapses are a little bit stronger and they may be firing a little bit more. Okay. Now I'm going to end with just a couple slides. I just have like 2 more minutes to show you this behavioral effect because I think it's important for what Ingrid and Connie and others have been talking about these different phenotypes. So this alpha-1 mutant mouse has very poor cognitive function. I mean, it's hard to call a mouse stupid, but this mouse, if you try to make this mouse solve problems, it cannot do it, which is remarkable given that there's no change in SYNGAP protein, and 75% of the SYNGAP proteins are unperturbed. Only this alpha-1, which is maybe 25 or 30%, These animals fail in mazes where they find their ways around, and they're also more susceptible to seizure, okay? But remember, they didn't have any changes in dendritic phenotypes, right? Like the regular heterozygous mouse. And I should say that all the behaviors I'm going to show you are all very, what are called penetrant in the heterozygous SYNGAP knockout mouse. So both of these you would see in the SYNGAP heterozygous mouse. These innate behaviors, which are less dependent upon learning and memory and synapse plasticity, and are related more to anxiety and innate fear, these behaviors are majorly disrupted in SYNGAP heterozygous mice, but are perfectly normal in these PBM mice. So that tells us right there that we have a brain-wide systems level behavioral dissociation between one of the isoforms potentially and the others. And then this is something I really want to show because there's been a lot of discussion about how much SYNGAP do you need to, so in the Syngapians, how much more, say somebody comes out with a therapy that raises SYNGAP by 20%. Is that enough to improve? Like, it'd be nice to see some evidence in that. And so we made another mouse. This is again published in this paper earlier this year, where we replaced beta subunits, these dendritic isoforms, with synapse isoforms, alpha 1 and alpha 2. We couldn't surgically do just alpha 1, so hopefully maybe Rick's lab will be able to figure out how to do just alpha 1, but what we were able to do is replace betas with alpha 1, alpha 2. And it looks like in these mice, we raised by 30% alpha 1 and alpha 2 at the expense of beta. So total SYNGAP is exactly the same. It's just you lost the betas and you give them more alpha 1, alpha 2. And in these mice, what we find is actually they're smarter. So just a 20 or 30% increase in just the alpha 1, alpha 2 protected them from seizure. So they took them much longer to seize in a seizure test, and also made them learn this maze that their cousins that had the disrupted alpha 1 failed miserably. And the important thing is that this experiment works in a wild type mouse, but it also works in a SYNGAP heterozygous mouse. So we can cross this mouse to the existing SYNGAP heterozygous mouse that has behavioral deficits, has learning deficits, has seizures, and what we show is that this raising alpha 1 by 30%, roughly 30%, protects them from seizure and also improves their memory. Okay, so in conclusion, protein isoforms clearly have unique molecular functions, and they have functions within different compartments within neurons, and almost certainly, based on this 2 areas of the cortex, they have different functions in different parts of the brain. Okay? And then to the encephalopathy mechanism that we talked about this morning, the way we conceptualize this is it may not always be this clean, but different when you lose these different isoforms in SYNGAP1 heterozygosity or haploinsufficiency, we think that different phenotypes or outcomes in the encephalopathy can come from the loss of different isoforms. So I'd like to thank everyone that funded the lab, including SRF, Leon and friends, lots of NIH, and of course, all of our collaborators. Thank you.
He was my postdoc, but he's only getting 2 questions.
I'm not sure I got it all, but I tried hard. How much do these isoforms differ according to different neuronal cell types?
Fantastic question. So we're currently doing that. So we're doing single cell long transcript sequencing to be able to do this. And so SYNGAP itself, if you just look at all the transcripts, has a very dichotomous single cell profile across the brain. Okay. I would bet, I don't know, a year's salary that we're going to see the same granularity, perhaps even more so in the isoform expression. So, and that will be the future of the work because we'll be able to get more specific with connectivity and circuits.
Gavin, that was a great talk. You had me at potassium channels.
I knew you'd be the most interested one.
So I have to ask, are you dissecting out, these neurons have dozens of potassium channels, obviously, and KV4.2 was a tool.
Yeah.
So are you trying to dissect out exactly, and are you thinking it's going to likely be one, or it's going to be multiple? Yeah,
so one day I'll give the full talk to you, and it's out for, we just submitted it, and it'll be on bioRxiv soon. But for this first paper, we know IA currents are disrupted, all right? which is in these neurons mostly Kv4.2. But the nice thing about dominant negative Kv4.2 is it's promiscuous, so it'll bind up to lots of different Kvs. And so it's a generalized potassium channel tool. What I'll say is that Kv4.2 isn't really associated with resting membrane potential, but these neurons are also very hyperpolarized. And so certainly multiple potassium channels are disrupted. And we're currently, David Ginty just published a paper a few years ago showing this very nice, using a different series of toxins, you can dissect all these different potassium channels. We're doing that right now. Yeah.
All right, great. I think we better move on. Thank you, Gavin.
Yeah, thank you.